Rare, precious, highly valued, emerald. It is the most famous and valuable green gemstone. Its beautiful green color, combined with durability and rarity, make it one of the most expensive gemstones. Tons of rock need to be unearthed to find such a precious commodity, and from a common rock, it is polished to reveal its inner beauty and brilliance. To possess it connotes a singular achievement. No wonder that this exquisite gem represents a milestone of immense significance. 55 long and eventful years. Much like the coveted gem, is the Philippine Association of Medical Technologists, or PAMET. Honed through challenges, polished through time. Today, representing a valuable resource, serving as a significant partner in the delivery of quality healthcare in the country. As it celebrates 55 years of meaningful transformations, PAMET has achieved a status akin to emerald. Precious, coveted, and shedding its brilliance towards the development of world-class laboratory professionals. In the late 16th century, the Spanish Empire had occupied Luzon and established Manila as their capital. Along with the Spanish occupiers came the members of religious orders aiming to spread the Catholic faith to the archipelago. These various religious orders established hospitals for the poor, and institutions of higher learning for the island's population. In 1887, the Spanish colonial authorities established the Laboratorio Municipal de Manila for the laboratory examination of food, water and clinical samples. It was the forerunner of the modern laboratory in the country. One of the well-known historical figures of the said laboratory was the Philippine war hero General Antonio Luna, who was employed in 1894 as a chemical expert, and pioneered environmental studies, water testing and forensics in the Philippines. During the American occupation, the Philippines had its first most advanced laboratory. The biology laboratory of the Bureau of Science, built in 1904, was used for the investigation of causes, pathology and methods of diagnosing and combating human diseases. The Philippines was making inroads in the delivery of healthcare services. The advent of World War II, however, decimated the said advances. From the ashes of war, the remnants of the laboratory left by the United States Army started as the seeds towards the modernization of the Philippine medical technology profession, guided by Filipinos themselves. Fueled by the desire to uplift the profession and enhance its recognition, PAMET came into being in 1963. Holding its very first annual convention in 1964, 55 years ago today. In just a short span of 5 years, its efforts paid off, with the Philippine Medical Technology Act enacted into law. By 1973, PAMET was officially recognized as the only accredited professional organization of registered medical technologists in the Philippines. So many illustrious names had added sheen and brilliance to the practice of medical technology in the country. From pioneers like Dr. Pio de Rota and Dr. Prudencia Santa Ana, to the first PAMET president, and to the successive PAMET presidents who rose to the challenge of their times. Each working tirelessly, as PAMET expands to other parts of the country, through the formation of chapters in various provinces and locales. Professionalizing its ranks, introducing innovations to modernize the profession, lobbying the government to accord medtechs the recognition and importance they deserve, positioning medical technologists as partners of health and hygiene of local communities, raising the profile of medical technologists as among the best in the world. 55 years of painstaking work, 55 years of enhancing its reputation, 55 years of building one success into the next. 5.5 decades of service to Filipino medical technologists. Today, the Filipino medtech has emerged as an indispensable pillar in Philippine healthcare, bound together by the common vision of sustaining excellence, as they look forward to inspiring the future generations. A rare breed, a sought-after expert, a precious resource, like emerald. This is the Philippine Association of Medical Technologists. PAMET. Sustaining excellence. Inspiring the future.
God, who by calling us to the vocation of a medical technologist, has placed upon us the obligation of being a constant help in the scientific care of the sick. Grant us by thy divine light a deep insight into the serious responsibilities of our task. By thy divine wisdom, awaken in us a growing zeal and determination to increase our knowledge of how to search for the underlying causes of sickness and disease, how to recognize the evidence of physical changes, how to make important chemical analyses, and other valuable tests so helpful in caring for the sick. By the divine love, permit us in this way to share with those who directly care for the sick, that thus we may be constantly working through the eternal physician, Christ our Lord. Amen.
Ang Pilipinas na ang may pinakamataas na kaso ng COVID-19 sa Southeast Asia. Mabilis ang pagtaas ng bilang, kung kaya't mas maaksyon ang eksena sa mga ospital. Bukod sa mga doktor at nurse na nangangalaga sa mga pasyente, mabigat din ang pakikipagbaka ng mga medical technologists, ang ating mga bayani sa loob ng laboratorio.
Ang mga medtechs natin, ang siya din nag-examine ng mga iba't ibang body fluids nanggagaling po sa katawan ng tao, mga PA, mga patuli, rema, siyempre po ang dugo, no? At saka yan nga pong mga swab specimen para po sa pag-examine ng COVID-19. Sila po ang may pinakamalaking risk pagdating po sa exposure, hindi lang naman ang pagharap sa mga pasyenteng posibleng kami mismo mga medtech ang humaharap sa virus. No? So, face-to-face po yan kung tutusin. Virus versus medtech.
Bukod sa mabigat na pressure sa trabaho, dagdag pa sa hirap ng mga health workers ang nararanasang diskriminasyon mula sa mga taong kanilang pinaglilingkuran.
Marami sa ating mga health professionals, kasama ng mga medtech, ay pinagdidirihan na ibang tao. Marami po sa kanila ay ayaw patuloyin sa kanilang tinutuluyang mga apartment or subdivision. At meron din po na hindi pinapayagang pumasok sa iba't ibang establishment. Ang mga frontliners po, kasama na po ang mga medical technologists, ay siya po natin kakampi. Ang ating pong kaaway dito ay ang COVID-19 virus.
Buwis buhay man ang napiling propesyon, walang pinagsisisihan si Philippine Association of Medical Technologists o PAMET President Ronnie sa kanyang trabaho.
Naniniwala ako na isa ito sa mabisa at magandang paraan upang makatulong sa ating mga kababayan at upang magamit din natin ang ating mga kaalaman tungkol sa siyensya. Kaya with or without COVID-19, I will be forever medical technologist.
Para kay Ronnie, ang pagsubok na ito sa buong mundo ay nagbigay rin ng pagkakataon upang ating mapagnilayan ang mga bagay na tunay na mahalaga sa buhay.
Na-realize natin na life is so short. Napakaigli talaga ng buhay. Sa isang iglap ay pwedeng mawala ang lahat. At siguro mahalaga rin na ma-realize natin na ang lahat ng meron man tayo sa mundo ay nabibilang sa Diyos. At the end of the day, masasabi natin na only the essentials matter most. Una-una is our faith. Hindi dapat mawala ang ating paniniwala at pananalig sa Diyos. Pangalawa na masasabi kong essential ay ang ating pamilya at mga kaibigan. At syempre, realization din na hindi tayo dapat magpabaya sa ating kalusugan at sa ating kaligtasan. So, let's all fight this COVID-19 together. Mananagpasan natin ito.
Before we start our webinar, here are some reminders and guidelines for our virtual participants. First, please observe proper webinar and video conferencing etiquette. In addition, please change your settings to speaker view. Also, you may post your questions through the link shown in your screen. Alternatively, you can also post your questions through the chat box in your Zoom console. Lastly, details on how to get your e-certificates will be flashed at the end of this webinar. We expect your adherence to these guidelines. Thank you very much. Dear colleagues in the profession, the Philippine Association of Medical Technologists warmly welcomes you all to our webinar series. This three-part webinar is focused on enhancing the competencies of medical technologists on quality RT-PCR testing for COVID-19. Allow me to introduce our moderator for this webinar. She is the chair of the PAMET Committee on CPD, a former national president of PAMET, and member of the PAMET Advisory Council. Ladies and gentlemen, Dr. Leila Florento.
Good evening, everyone. I will be the moderator for this first live webinar of PAMET. To start, let me introduce our president, Mr. Ronaldo Puno, to give the opening remarks.
Good evening. Can you see me? Good evening, good morning, and good afternoon to all our colleagues. Can you hear me?
Yes, sir. Yes, sir.
Okay. So, again, good evening, good morning, good afternoon to all our colleagues, wherever you may be, because we are joined by participants from different parts of the Philippines as well as in the world. Today is another milestone for PAMET as we offer this very first webinar series entitled Enhancing the Competencies of Medical Technologies for Quality RT-PCR Testing for COVID-19. And we are very glad with the overwhelming response from you, our dear colleagues, that when we opened the registration, we had to close it in less than 24 hours. Of course, that is due to the heavy influx of interested health professionals. Right now, we have around 1,000 registered participants, with more joining through our Facebook live streaming. And we apologize to those who were not accommodated by the platform, of course, due to some limitations. And I would like to extend our deepest appreciation to our CPD committee led by Dr. Laila Florento, the e-learning and social media communications committee led by Mr. Oliver Dumawal, to Ms. Geraldine Dayrit, the main proponent of this virtual seminar, for coming up with this innovative approach in our desire to continuously deliver relevant continuing educational activities such as this one, despite the difficult situation that we are in. Of course, special words of thanks to our invited speakers. Most of them are from the United Kingdom, who, despite the distance, willingly agreed to share their knowledge and expertise, and they will be properly introduced later on. I'm sure that we are in agreement that COVID-19 pandemic changed and continuously changing our lives. What we are experiencing now is unprecedented, and the challenges that it brought to us is really overwhelming. And it is a time where the knowledge and expertise of medical technologies are needed and highlighted more than ever. It is in this context that we decided to provide a platform for our professionals to learn more about the novel virus, as well as to equip them with important competencies in dealing with the pandemic through scientific laboratory techniques such as PCR. And we are hoping that the three sessions that we have prepared and happening in 3 different days will upgrade the level of confidence of our frontliners who continuously fight the invisible enemies every day when they enter the laboratories. And in closing, I would like to commend the immeasurable contributions and heroism of all our medical technologies. I hope that this session will provide us with the necessary knowledge. And because without the medical technologies, quality, timely, and accurate laboratory results cannot be assured. Again, enjoy the session, and together, let's all fight and beat COVID-19. Good afternoon to everybody.
Thank you very much, Mr. Puno. Now we'll start with our first speaker. Our first speaker is a graduate of BS Medical Technology, a registered medical technologist, Masters of Science in Microbiology from University of Santo Tomas, with Diploma in Research and Development Management from University of the Philippines and Diploma in International Graduate Program of Veterinary Science and Technology from Chulalongkorn University, Thailand. Currently, she's undergoing PhD in Biological Sciences Molecular Track at University of Santo Tomas, and at the same time PhD in Veterinary Science and Technology at Chulalongkorn University, Thailand. She was formerly a lecturer at UST, assistant professor and chief medtech in institutional ethics reviewer at Trinity University of Asia, and medtech at Film Life Medical and Laboratory Services. Currently, she is a research consultant, assistant professor at Family Clinic, special lecturer at Graduate School, Trinity University of Asia, assistant professor at Department of Medical Microbiology at College of Public Health, UP Manila, and is a volunteer for COVID-19 RT-PCR testing, Bayanihan to Heal as One Act. She received several awards and international travel grants, the most recent of which is Chulalongkorn University Doctoral Scholarship. She had several research publications, paper and poster presentations, a patent from the OST on production of mordant from ipil-ipil, and many other collaborative research works. Colleagues, let us hear from our first speaker, Geraldine Bodomo Dairit.
Okay, can you hear me?
Yes.
Thank you very much, Dr. Florento, for that warm introduction. A pleasant evening, morning, and afternoon to all the listeners of this PAMET free webinar series. From what I have heard, this is, we are earning our CPD units in the comfort of our zones. On a positive note, this is the new normal. Tonight, I will be sharing to you my presentation. Okay, let me see if I could. I'm having a technical problem on sharing my slides. Let me see. Okay. Okay. It's in Zoom. Okay. Can you see my slides now? Am I sharing my slides now?
Not yet, Ma'am Gigi.
Not yet. Let me check. This one.
Okay, good.
Okay.
It's okay now.
Slideshow.
It's in slideshow. Okay. From what I understand, we will be earning our CPD units in the comfort of our zones. On a positive note, this is the new normal. Tonight, I will be taking you to a new dimension to have you understand that we are currently in a war against a virus, the SARS-CoV-2 virus. The ongoing situation poses a challenge for laboratories as the outbreak is widespread. In order to win the war against the virus, we have to do planning. We must prepare for the battle and try to dissect the enemy. To prepare for the battle, we have to know and understand our current situation, assess our capabilities, and recognize our limitations. We have to know the guidelines and protocols set by the World Health Organization, Disease Control and Prevention, Department of Health, Research Institute for Tropical Medicine. We also have to determine the setup of the lab and identify those materials and equipment dedicated for a specific area. We have to apply a unidirectional flow in molecular settings and boost resistance particularly the biosafety measures in the laboratory. We have to know our enemy. That is why later, after my topic, we are going to understand the background of the virus by knowing the basics of molecular biology, virology, and the method, how to expose these viruses' identity. The next slide will reveal to us our current situation. Coronavirus disease 2019 is affecting people around the globe. It is caused by an infection with the causative agent, an RNA virus, which is the severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. It was first identified in Wuhan, China in December 2019, thus the term COVID-19. DOH COVID-19 Bulletin number 69 as of May 22 shows 163 additional confirmed cases with positive test results, bringing the total number of affected Filipinos to 13,597, with 92 individuals recovered from the disease and 11 deaths reported. Increasing the nation's testing capability would help in easy detection of infected people, prevention of further transmission of the virus, and confirmation of people without infection. As of May 14, 2020, there are 30 RT-PCR licensed laboratories, and that is equivalent to at least 90 RMTs. Why 90? DOH directed that there must be at least 3 registered medical technologists per COVID-19 laboratories directly involved in the testing. And as reported, there are 105 total number of laboratory applications. This means, let us do the math, that there would be at least 315 RMTs needed. The question is, can we answer to this call? In a nutshell, this slide shows the limits of our testing capacity. In relation to the 30 accredited labs nationwide, it was mentioned during the DOH Beat COVID-19 virtual presser, an average of 8,000 tests can be done per day. With a target of at least 1% of the 110 million Filipinos, that is 1.1 million, there were only 184,857 Filipinos tested, which is only 20% of our target. The usual turnaround time for the testing is 1 to 2 days, but in reality, this may extend for 1 to 2 weeks due to a lot of factors. Testing backlogs could be due to the tight capacity for the huge volume of samples received each day. Given all this information, now, the question is, are we really flattening the curve or just hitting the ceiling of our testing capacity? On this slide, I would like you to meet our standard setters. Let us meet the offices behind the policies and guidelines on COVID-19 laboratory testing. The Bayanihan to Heal as One Act, officially designated as Republic Act number 11469, which was enacted declaring the existence of a national emergency arising from the coronavirus disease 2019 situation. In our work, we make it a point that we are doing the standard and performing the best practices. This means adhering to the guidelines set by the World Health Organization and Center for Disease Control and Prevention through our Department of Health. Part why we are here is because of our aim to ensure safety of personnel and of the general public, as well as the quality and accuracy of the reports generated by these COVID-19 testing laboratories. The Research Institute for Tropical Medicine, or RITM, of the Department of Health has set operational guidelines and gives the proficiency tests for all personnel applying for this lab, while the Food and Drug Administration gives the list of approved COVID-19 test kits for commercial use. It is mandatory testing that lab shall use FDA kits registered by FDA, reagent, and devices. As medical technologists, we deliver the most accurate and precise results, and these are indicative of reliable and standard laboratory setup, organized and updated protocols, well-trained and highly competent personnel. As previously discussed, we have to follow the standards, criteria, and requirement prescribed in the DOH assessment tool for licensing. RITM's Biosafety and Lab Assessment Tool, the World Health Organization's Risk Assessment Form, plus compilation of different DOH issuances. Manual operations for standard operating procedures of the lab, which include the work instructions like receiving or rejecting of specimens, step-by-step procedure of inactivation, RNA extraction, and amplification protocols. These would be used as references and guide. Work instructions or manual of operations, rather, for biosafety and biosecurity policies and procedures, which we will discuss in brief in the succeeding slides. Specific guidelines in handling and transporting specimens will be discussed in the second session. Disposal of infectious wastes. Remember that these infectious wastes must be handled properly to protect the environment and human health. Ensure strict compliance with infection prevention and control guidelines within the facility. For records management, we follow the DOH Department Memorandum Number 2020-0110, giving directives to all hospitals, private or public, and healthcare facilities on reporting COVID-19. Lastly, preventive calibration of equipment must be done, as well as daily cleaning and maintenance must be documented. Let us take a look at the molecular laboratory. Minimum number of personnel shall be 7, of course, depending on the workload. We have 3 full-time analysts. These are the registered medical technologists, 1 pathologist, 1 full-time medical laboratory technician, 1 full-time receptionist, and 1 full-time encoder. The 3 full-time registered medical technologists shall have appropriate trainings on fundamentals of biosafety and biosecurity and proficiency in molecular diagnostic techniques. All these laboratory personnel should undergo fit testing for respirators, annual medical examination, plus influenza vaccination, and also continuing updates or trainings on biosafety, biosecurity, and new techniques. Laboratories shall have adequate and appropriate areas to safely, effectively, and efficiently provide services to the clients. There must be dedicated space for reagent preparation and storage, receiving of specimen, virus inactivation and RNA extraction, amplification using RT-PCR, clerical activities, which include receiving and compiling forms and result releasing. Adequate lighting in all areas. Split type aircon shall be installed, and the air direction should be away from safety cabinets. The layout of the facility that I will be showing in the succeeding slides is based on AO 2020-0014 Appendix D. This is the specimen reception area, Note that in this area, we handle and process specimen with suspected or confirmed COVID-19 infection. Negative pressure for areas handling live samples should have exhaust, producing at least 12 air changes per hour and directed away from people and adjacent structures. Standard equipment, but not limited to sensor, activated sink, refrigerator for reagents, working tables and chairs and storage cabinets must be in this area. Dedicated pipette tips, pipette and pipette tips must also be in place. As you can see, pass box is provided as control measure. Number one, it could be used like in receiving area for receiving specimen from referral hospitals or walk-in patients, and the other one at the middle left to transfer specimen to another room. Initial processing of all specimens should take place in an appropriately maintained and validated biosafety cabinet. And we have here biosafety cabinet class 2 type A2 shown in the picture. Also in this area is the availability of the use of appropriate disinfectants with proven activity against envelope viruses. Inactivation of the virus should also take place in an appropriately maintained and validated BSC. The external lysis buffer on the listed common RNA extraction kit is effective in inactivating the COVID-19 virus without heat or other additional means. Changing area is provided to highlight the importance of wearing PPE when handling and processing specimens. On this slide, we have the reagent preparation area. Note that MedTech in charge should have a prior idea of how many samples are to be processed for the computation and preparation of the master mix. Laminar flow to be used should protect the material in this case, the master mix. Positive pressure, which shall have filtered air supply with 90 to 95% efficiency, must be applied. This means that we are protecting here the master mix from contamination. Also, reagents and enzymes used in the preparation of the mix are sensitive to temperature and light. We need this PCR hood to protect the master mix from deterioration as well. Standard equipment, but not limited to sensor-activated sink, refrigerator, and freezer for reagents, minifuge, working tables and chairs, and storage cabinets must be in this area. Dedicated pipette tips, and microtainer tubes must also be in this place. Cold rack for the PCR tubes can be used to maintain the right temperature during preparation. As you can see, pass box is provided as control measure. This is placed to transfer the master mix to another room. In this case, the other room is the template adding room. No changing area for PPE is provided, but the use of lab coat, gloves, and mask is a must. This is the RNA extraction area. Note that the virus is already inactivated on this part. If not, appropriately maintained and validated BSC must be in place. Depending on our local protocol, some inactivates by heating the ali-coated sample. Please note that heating plate for inactivation must be inside the biocontainment facility available in this area. Negative pressure for areas handling live viruses should have exhaust producing at least 12 air changes per hour and directed away from people and adjacent structures. Extraction area must contain the standard equipment, not limited to sensor, activated sink, refrigerator for reagents, freezer for extracted RNAs, minifuge and microcentrifuge machines, working tables, chairs, and storage cabinets, dedicated pipette, and pipette tips, and consumables like containers, micro containers must also be in place. Template adding room is equipped with a PCR hood to protect possible contamination of the template and master mix. This also has its dedicated pipette tips, and minifuge for PCR tube spin down. As you can see, pus boxes are provided for control measure. These are placed to transfer the inactivated or samples from receiving room to extraction room, from extraction room to template adding room, from reagent preparation room to template adding room, and from template adding room to the amplification room. Changing areas are provided for each room to prevent contamination. This is the amplification room. For our RT-PCR room, exhaust must produce at least 6 air changes per hour, also directed away from people and adjacent structures. This area must contain the RT-PCR machine and computer for viewing and analysis. The machine must be backed up by a UPS in cases of power failure or electricity fluctuation. Changing area is provided for each room to prevent contamination. This area requires no PPE. But of course, we have to wear our lab coats. And clerical activities be done to compile all files and prepare all reports for releasing. Again, this layout was adopted from AO or Administrative Order 2020-0014 Appendix D1 with some modifications. Below is the unidirectional flow in molecular testing. As you can see, the flow is from clean to dirty, from master mix to extraction to amplification. This is showing dedicated spaces for different main activities. Different areas in these rooms are provided. Changing rooms are also highlighted to give emphasis to different PPEs for different activities. Pass boxes are available to maximize the workflow and minimize the use or unnecessary changing of PPEs. Other rooms are provided to maximize the efficiency of the personnel. To boost our resistance, we need to identify the hazard. So the hazard here is the laboratory acquired infection. The pathogen is the SARS-CoV-2 virus. And activity is through inhalation exposure to infectious aerosols. In the lab, what are the activities that would cause aerosolization? This would include but not limited to centrifugation, alicoating, pipetting, removing of caps or lids of containers in areas where we handle specimens and manipulate the live virus. Processing of respiratory samples for inactivation procedure and RNA extraction shall be performed in a biosafety level 2 facility, enhanced with biosafety level 3 equipment in a biosafety with biosafety level 3 equipment and practices. So definitely we can handle them using biosafety level 2 facility, which may include the enhancement includes directional airflow, the use of fit-tested NIOSH-approved filtering phase piece respirator, and others. So for control measures, each laboratory should conduct a local, that is institutional risk assessment to ensure safety of all the persons. All procedures must be performed based on the risk assessment and only by personnel with demonstrated capacity and capability and strict observance of any relevant protocols at all times. Control measures include information control, personnel, physical security, transport, emergency response, and environmental management. So we are now at the end of my slide. I hope we are all on the same page. So we have to win this battle against SARS-CoV-2. Our profession could make a difference. We could make a difference. We will win this battle. Thank you very much for listening, and I hope you learned a lot. Have a nice day.
Thank you very much, Ms. Gigi Dairit. We will have the questions at the end of the talk of all the speakers. Kindly prepare your questions and send them to the site. The next speaker, I will introduce the next speaker. He is a graduate of BS Biotechnology from University of Wrocław, Poland. He completed Master of Science in Translational Medicine, University of Helsinki, Finland. Currently, he is a PhD pathology candidate at University of Cambridge, UK. He received several scholarships and travel and conferences awards, the most recent of which is the PhD studentships from Department of Pathology, University of Cambridge. He presented papers and posters in different conferences in various countries. He was a visiting researcher at University of Edinburgh, UK, a researcher at University of Helsinki, Finland, and currently marketing advisor and team leader at Polygia, Cambridge, UK. At present, he is the laboratory project supervisor for natural sciences students in Cambridge. The topic he will discuss will be fundamentals of molecular biology and molecular-based testing. Let us now hear from our next speaker, Dr. Thomas Hendrik Benedict.
All right, hope you can all hear me. Thank you for the introduction, and good evening, everyone. Greetings from the United Kingdom. I'm gonna now share the slideshow with you. All right, so my name is Thomas Benedict. I'm currently in the final year of my PhD at University of Cambridge. My major interest in virology is to understand the atomic level of viral interactions with their host through biophysical and structural studies of viral proteins. As you will learn during this training, viruses are unique entities that not often follow the classical principles of biology, also pushing every naturally occurring mechanism to the far extremes. That's why they can be so dangerous, but also, in my opinion, super fascinating. During my talk, I will cover the basis of molecular biology, which many of you may be already familiar with. But at the end of my presentation, I hope all of you will know the differences between DNA and RNA and understand their basic processing. All right, I will start with asking a rather philosophical question. What is life? Well, this might seem a trivial one, but even the greatest minds have never been able to formulate a satisfactory definition of life. I, as a molecular biologist and biochemist, will now try to introduce at least a few conditions which I believe a living organism must fulfill. Okay. A living organism has to be structurally defined and highly organized. Even the simplest bacteria have distinct elements that not only look similar, but share the same functions. Those elements are further organized into more and more intricate systems, as represented in this figure on human body as an example. Moreover, all physiological processes of this complicated system are precisely regulated and coordinated in time and space. Here, many of you may recognize what appears to be a metabolic map, But in fact, this is a map of public transport system in London, which I think represents quite well at the macro scale how a living organism functions, with hundreds of routes, complicated timetables, while providing service to millions of individuals at once. And now it's the most important bit, without which the previous points wouldn't be possible nor make any sense, the ability to reproduce. The first and in fact only goal of life is to keep living. This can be achieved by saving all necessary instructions in a code that can be passed on to the next generations, which will look and function the same as did their parents. Such code is the DNA code. It alone includes all necessary information for the organism to develop, function, and further reproduce. This code is universal across all living organisms, whether it is a bacterium from millions of years ago, a tree in your garden, or your best friend, they all use exactly the same genetic alphabet. For what it does, it is actually very simple. It consists of only 4 basic elements called nucleotides, also known as bases. Just imagine our alphabet having only 4 letters and yet being able to contain more information than Wikipedia does. So each nucleotide, thymine, cytosine, guanine, and adenine, is a chemical molecule of slightly different properties, like 4 blocks of different shapes and colors. Those bases are arranged in a very long sequence, and that very DNA sequence determines all characteristics of each individual. As an example, human DNA sequence consists of over 3 billion of these bases. To form such long molecule, it requires a strong backbone. And in DNA, that backbone is built of the sugar called deoxyribose. One of the key characteristics of nucleotides is formation of complementary pairs. Nucleotides are unhappy alone, but also they have their favorite companion. And so T for thymine always binds adenine, whereas cytosine pairs only with guanine. Now I'm going to talk about probably the most important process in molecular biology, replication. As I just mentioned, nucleotides like being in pairs. Therefore, DNA is in fact a double-stranded molecule, and this makes it able to generate its own copies in the process of replication. So replication requires perfect precision, as we don't want to introduce any errors to our valuable instruction. We also need new copies of this long sequence in a relatively short period of time, which in turn requires high processivity. To meet those 2 conditions, nature came up with a very robust machinery, which I am now going to briefly introduce. First of all, these are the elements that we need: DNA template, 2 enzymes, primase and DNA polymerase, and a pool of 3 nucleotides. Our complete DNA code, also known as genome, is extremely long. So to protect it, it is densely packed inside every cell. As it is unfolding, we can eventually see the double-stranded elongated molecule formed by the paired nucleotides, and those can be further separated into 2 chains. Each chain is now serving as a template for specific enzymes that can regenerate a double-stranded molecule based on the complementarity of nucleotides. During this first stage, initiation, the enzyme called primase starts replication by synthesizing the first short string of complementary nucleotides known as primers. In the second stage, another enzyme, DNA polymerase, binds each primer and continues the DNA synthesis until the entire sequence is duplicated. This stage takes the longest and it's called elongation. Throughout this process, a pool of 3 nucleotides must be available so the enzymes can incorporate them into the growing complementary strands. Once the DNA molecule has successfully undergone replication, we reach the final step, termination, when the enzymatic machinery dissociates off the DNA, leaving two identical copies of the genetic information. As depicted by blue and red, each of them consists of one newly synthesized strand and one from the original DNA template. Let's briefly summarize what we have learned so far. DNA information is present in almost every cell of our body, stored in the nucleus. It contains the complete set of instructions, which is called the genome. Thanks to replication, all that information can be duplicated and passed on to the next generation of cells. But now, how can the cells actually read and use this instruction? Well, this phenomenon is called gene expression and requires two steps. Transcription, leading to conversion of DNA into RNA, and subsequent translation, where the RNA is used by ribosomes to synthesize proteins, the final product of gene expression. It is important to bear in mind that in the given moment, every cell needs only small parts of their genome, called genes, to be expressed. Cells can also tightly control the strength that each gene is expressed with. To achieve that localized and timely controlled expression of the long DNA molecule, cells can select which genes to express and turn their corresponding DNA fragment into a short, more transient carrier of genetic information, RNA, in the process of transcription. So now, let's have a closer molecular look at transcription. Similar to replication, transcription machinery is present in the nucleus and requires several elements such as template, the enzyme called RNA polymerase, and a pool of free nucleotides. In the first step, the densely packed genomic DNA, our template, again needs to unfold to give access for the enzyme. However, unlike replication, where the entire DNA gets copied, here we need only a short and very specific fragment to be transcribed, a gene. Also, in contrast to replication, this process doesn't require primers. Hence, only one enzyme, RNA polymerase, is sufficient for the complete cycle. The RNA polymerase can distinguish between the two complementary strands and transcribes only the one labeled here as antisense strand, effectively synthesizing a faithful copy of the sense strand. It is important to remember that by convention, when presenting any DNA sequence, we always refer to the sequence of the sense strand. Once the transcription has terminated, the nascent RNA molecule leaves the nucleus and becomes a template for protein synthesis in the cytoplasm. There are three major structural differences between RNA and DNA. The sugar scaffolding is ribose instead of deoxyribose. RNA molecules are single-stranded, and thymine is replaced by another very similar nucleotide, uracil. Transcription takes place in the nucleus, but once the RNA molecule is ready, it enters the cytoplasm where serves as template for protein synthesis carried by the ribosomes. As I mentioned at the beginning, viruses are peculiar entities that rarely adhere to the principles of biology. Many viruses, including the novel coronavirus, keep their genetic information as RNA, which can get directly translated into proteins. Other RNA viruses, such as HIV, want their RNA genome to become more stable and stay with the host for longer. Therefore, use a mechanism that is otherwise very rare in living organisms, reverse transcription. In this process, the viral enzyme called reverse transcriptase synthesizes a double-stranded DNA using the viral RNA as the template. This process also requires primers for initiation. This is a very important reaction, often used in molecular often used in molecular biology, and an essential diagnostic tool for detection of viral infections, which you will hear more about in the coming talks. In my final 2 slides, I will summarize the major differences between DNA and RNA and the main processes they are involved in. Both DNA and RNA use the same building blocks, except for thymine, which in RNA is replaced by uracil. The backbone sugar for DNA is deoxyribose, whereas for RNA it is ribose. DNA is double-stranded and RNA consists of only one strand. DNA encodes the whole genome. RNA corresponds usually to only one gene, but keep in mind the exception of some viruses. DNA you will find almost exclusively in the nucleus. RNA, in turn, once ready, goes from the nucleus to the cytoplasm. Then DNA is very stable. Pure DNA in solution can be stored at room temperature for a very long time without much damage. RNA, in turn, is very unstable and prone for enzymatic degradation. Therefore, when working with RNA, you need to be super careful and keep organized not to lose your sample. Now, looking at replication versus transcription, both take place in the nucleus. During replication, we synthesize more DNA from DNA. In transcription, we turn DNA into RNA. Replication has excellent fidelity. We don't want to introduce any errors to our valuable genetic instruction. Transcription, in turn, generates a transient product. Therefore, it is more forgiving and occasional errors won't have a big overall impact. Replication isn't strand-specific, it is bidirectional. Both strands get replicated simultaneously. Transcription uses only the antisense strand. Only replication requires primers, and replication, unlike transcription, is not gene-specific. The whole DNA sequence gets replicated. And finally, both processes are tightly regulated and very complicated. All right, so that was me. In the next talks, you will hear more about different molecular techniques that utilize those natural processes. They are very important, both in research and diagnostics. Thank you so much for listening.
Thank you very much, Thomas, for that very detailed explanation about basics in molecular biology. Now we go to our third speaker. Our third speaker is a graduate of BS Medical Technology at Martinez College, Manila, completed Master of Science in Public Management in Development Academy of the Philippines, and Master of Science in Molecular Biology at Universidad Autónoma de Madrid, Spain. He finished his PhD Public Administration and PhD major in Molecular Biology and Molecular Virology at Universidad Autónoma de Madrid, Spain. His postdoctoral research fellowship were at London School of Hygiene and Tropical Medicine, UK, and Division of Virology, Cambridge University, Cambridge, UK. He presented several papers in different scientific meetings in various countries. He published papers in molecular biology and molecular virology in different peer-reviewed journals. He was formerly employed as bacteriologist at NRL for infectious diseases at DOH Manila and was formerly a board of director of PAMET as well. Currently, he is holding a post as Senior Executive Officer, Healthcare Scientist Team Manager at the National Infection Service UK, National External Quality Assurance Service of Public Health in England, UK. His topic for today is about fundamentals of molecular virology. Let us all welcome Dr. Teodoro Fajardo Jr.
Hello. Good morning to UK and good afternoon and good evening to the Philippines. Thank you, Dr. Laila, for the introduction, and thank you to the previous two speakers, Gigi and Thomas Benedict, about the DNA and the setup of the laboratory. I'm now going to share my screen. All right. Okay. Okay. So this is an introduction to molecular virology. Let me know if you don't see my screen. Is it okay?
It's okay, Doctor.
All right. So introduction to molecular virology. I know that most medical technology courses, they don't focus on studying virology. It's not that important before. Now it became very important. So now that's the purpose of this seminar. And I would like everybody to understand the basics of it. So let's start with the question. I cannot see my screen. Okay. Hold on. So what are viruses? That's the basic question. So just to start, we are all part viruses. So viruses are all around us. They are in the food we eat. Plants have so much viruses in there that you can actually eat them and they won't infect you because we don't have the receptor for those viruses. Only plant cells have the receptors. For example, the orange that you eat or the rice that you eat. And the water that you drink, or you swim in the river, you swim in the sea, they are full of viruses. So you can actually get them and without having a disease because you don't have the receptor. Okay. So another information is if we stretch all viruses on Earth, which is 1 times 10 to the 31, and we laid it end to end, it can stretch up to 100 million light years. The distance of 1 million light years is the distance of a light traveling in 1 year. Okay, so that's how many viruses are in the Earth. So a single bacteria can be infected by hundreds of thousands of viruses, and this virus is called the bacteriophage. So this bacteriophage, they infect some certain bacteria and inject their genome or nucleic acid content, as you can see in here, into the bacteria and start replicating. As Thomas said, the viruses, they don't qualify in the principle of life. They are non-living, so they require a live host to replicate. So another info is that a portion of the virus genome is actually in our genome. So we are part virus in our genome. So how do they do it? So in the beginning of life, we are constantly infected by a virus called endogenous retroviruses. These retroviruses, they require inserting their genome into the DNA inside the nucleus. So they cannot replicate by themselves. They have to insert their genome or a portion of a gene in the DNA on the nucleus, and the DNA will start the process of replicating our DNA, and in the process they also replicate the DNA or the gene of the RNA of the virus. So some of this RNA stays in the DNA of our cell. So this happened over a long period of time, and we actually carry them up to now. So this is actually the reason why some researchers say this is the cause of some cancers, because this genome could be activated and cause some cancers. This is under study, but this is one postulate. And another fact is 16 million people die of infectious diseases every year, and a large proportion of because of virus infection, influenza, HIV, hepatitis B, dengue, Zika, whatever. And 500 million for example, 500 million people are infected with chronic viral hepatitis. So COVID-19 disease, which is caused by SARS-CoV-2 virus, has killed so far 340,000 from January to May. So compare those figures. So what actually is a virus? So this is the big question now. Okay, a virus is a protein material. It's actually a molecular protein material programmed to invade the cell, which is the attachment, replicate, and then make progeny viruses. So hold on, I cannot. Hold on. Yeah. Okay. Can you actually see this screen that is blocking my it's blocking it. How do I? Sorry. Anyway, so that's the virus genome. So a complete unit of a virus is called a virion. So a virion is with a protein coat, which is called the envelope, as you see here in red. And an envelope is typically a bilipid envelope for an envelope viruses. Under the envelope is the membrane, which is supported by the envelope and the envelope protein, and it's attached to the to that envelope is the membrane, which you can see in here, the thin red line. And then under that is a viral capsid. It's another protein coat layer to protect the genetic material that it hosts inside. So the genetic material could be, as Thomas said, is RNA or a DNA. So it depends on the coat of the virus. We can call them envelope if they have the envelope. There's a coating, protein coating outside. If they don't have that, it's called non-envelope viruses. So this envelope are actually produced by the virus itself, and also some part of this envelope is from the envelope from the cell that they infect from the host cell. It could be from the endoplasmic reticulum, Golgi apparatus, or in the cellular membrane envelope. So the virus are non-living organism and only replicate inside the living host cell. So this is what Thomas emphasized in the beginning, the why it is life. So bacteria, mammalian plant, parasite, or amoeba have all viruses. So but not all viruses are harmful. Some help scientists in solving medical problems. And one of these is a good example are the bacteriophage. The bacteriophage, they infect bacteria, especially the drug-resistant bacteria, and they kill them. So it could be used in the future as a nanotechnology to kill drug-resistant bacteria. And also we use viruses for developing vaccines as a vehicle for developing vaccine. For example, in the coronavirus SARS-CoV-2 vaccine development, they have identified the gene that is antigenic. They get that gene and then insert that into a virus that is non-pathogenic because they took out the genetic material of that virus and replaced it with the gene of the SARS-CoV-2. That is, for example, adenovirus. So we can be infected by the adenovirus without the genetic material, complete genetic material inside. This will not replicate, but will boost the immune system to recognize the protein of that virus. For example, for now, SARS-CoV-2. So in naming the virus, there's a virus taxonomy. So there are many types of viruses, and actually they are named according to places where they are discovered. For example, Ebola from the Ebola River, or the Zika virus, which is discovered in Zika forest in Uganda, or the disease that it causes, for example, HIV, human immunodeficiency virus, hepatitis virus, dengue hemorrhagic fever virus, or a country or geographical region originally discovered this virus. For example, Middle East respiratory syndrome virus, or Japanese encephalitis virus, West Nile virus. But however, there is one thing that a misnomer, like Spanish flu that started in 1918 did not originate from Spain. It's actually Spain started the reporting of the Spanish flu. So it's stuck in their mind that it originated from Spain, but it's not. Okay. Viruses now, however, of all this, viruses are named now based on their genetic structure to facilitate development of diagnostic tests, vaccines, and medicines. So according to the new rules, no names of places or persons should be used to name a virus to prevent a dilemma or a brand of that place or a person. So in the case of a coronavirus, we start with the order. They belong to the Nidovirales. The family is Coronaviridae, subfamily Orthocoronavirinae. And the genus is Beta coronavirus, along with other like Alpha coronavirus, Gamma coronavirus, and Delta coronaviruses. And subgenus is Sarbecovirus, and the species is SARS-CoV-2 virus. Another species is the MERS. Another species could be SARS-CoV, and another one that causes common colds. So how viruses are characterized. Hold on. So they are characterized or categorized according to the nucleic acid that they contain inside their genome. So they could be a DNA viruses, and these DNA viruses could be a double-stranded DNA or single-stranded DNA. The single-stranded example of a single-stranded DNA is a parvovirus that infects animals or also human. And the double-stranded DNA virus are herpes, pox, and hepatitis B. So you can see here the examples of these viruses. And RNA viruses are could be single-stranded RNA or double-stranded RNA. One of the single-stranded RNA positive sense is the SARS-CoV-2. The coronaviruses are single-stranded RNA positive sense. Positive sense meaning when they infect a cell, they release their genome and proceed to translation already. So infection is fast, very fast. Virus enters the genome, the cells, the host cells, release its content, which is a positive sense RNA, and then translated into protein and starts infection. So that's how fast the virus is. Another one is a dengue, norovirus, Zika, hepatitis C, and HIV. So single-stranded RNA negative sense are Ebola, rabies, mumps, and influenza. And the example of the double-stranded RNA are rotaviruses, viruses, and African horse sickness virus. They can also be categorized or classified according to the coating that they contain. So there could be envelope or non-envelope viruses. So envelope, good example is the SARS-CoV-2, dengue, HIV, HCV, and influenza. And non-envelope or we call this non-envelope naked because they don't have the envelope covering, and they only have this covering for their genetic material or the genome, which is the capsid. And the example of this is the norovirus, rotavirus, and poliovirus. Also, another one is based on their morphology. So we can say coronavirus is a icosahedral virus, a positive sense polarity RNA virus. So it could be icosahedral like rotavirus, helical like the Ebola virus, and the complex like the bacteriophage, as you can see in here. And another classification are the size of the viruses. There are very big viruses that you can actually see in your gram stain. So these are called the megaviruses, the Pandora viruses, or Mimi viruses. So these Mimi, because they mimic the bacteria, because you can, the history of this is they can actually see it in the gram stain, but they don't know that it was a virus. They was mistaken as a cocci. So it was only in 2013 that it was discovered as it was a virus. So it's called Mimi virus. And then the smallest one are the circovirus and picornaviruses for small viruses. One good example of the picornavirus is the foot and mouth disease virus that causes the foot and mouth disease. And the polioviruses, of course, causes polio. So what is the SARS-CoV-2 virus? The SARS-CoV-2 belongs to the, as I said, it belongs to the family Coronaviridae, and there are subgenus this called alpha, beta, gamma, and the delta. The alpha and beta are only pathogenic to mammals, and gamma mainly causes bird infections. The 7 types of human coronavirus that causes human respiratory disease. Two of these are causing common colds, and 2 also causes other respiratory viruses, respiratory diseases. and 3 are the one that has causing problem recently. It's well, MERS-CoV 10 years ago, SARS-CoV like 20 years ago, and then SARS-CoV-2 just right now. The current coronavirus is SARS-CoV-2. The name of the virus, let's be clear on this. The name of the virus is SARS-CoV-2, and the disease it caused is COVID-19, which stands for Coronavirus Disease 2019. The old name Wuhan nCoV is obsolete now. You don't use that anymore because the WHO has renamed it as SARS-CoV-2. So when you say I am infected, you are infected with the SARS-CoV-2, you are not infected with COVID-19. You have COVID-19 disease. Okay, so let's go now to the SARS-CoV-2 morphology. So as I've said, the SARS-CoV-2 is an envelope virus, is spherical of about 120 nanometer in diameter. This is the graphical representation of the coronavirus. It has, as you can see, it has an E protein, which stands for the envelope, S protein, which stands for the spike, and M protein, which stands for the membrane envelope. The E envelope is the envelope that is synthesized, and also, as I've mentioned earlier, is a lipid bilayer, and it's very sensitive to desiccation and also to heat and the surfactant. The surfactant are the soap that we use. So these are anionic detergents that we use to disinfect our hands. So the envelope protein is very sensitive to this because of the bilipid layer on the surfaces. They're also sensitive to pH and alcohol and other virucidal agents. Okay, this makes these viruses, because of this envelope, this makes these viruses easier to sterilize than non-envelope viruses, such as HIV, dengue, hepatitis C. They can be easily sterilized as compared to the non-envelope, which is rotavirus or norovirus. Even if you use other virucidal agents, they cannot be easily killed. They can stay for a long time, like for months. But SARS-CoV-2 can only stay for days. And these viruses, the envelope virus like SARS-CoV-2, are sensitive to drying due to the envelope that I'm saying. And that is the reason why these viruses are easily spread on moist or wet conditions, like aerosol, droplet nuclei, moist surfaces, and moist tissues. So they like wet surfaces because they are very, very sensitive to drying. And they are easily killed by UV light. So in the lab, we kill the virus inside the biological safety cabinet by turning on the UV lights, because these viruses are very sensitive to this light. They destroy the genomic contents of the viruses. So inside this virus is a capsid. So there is a capsid inside. We cannot see it now. So the capsid is more rigid compared to the capsid to the non-envelope viruses. And the capsid, as I've said, is resistant to solvents, detergents, and ordinary disinfectants. These viruses, like I've said already, so rotaviruses and noroviruses. So the spike protein on the surface of the coronaviruses gives its characteristic name, corona, because it's shaped like the corona of the sun. It's not like the corona that you wear on your head. So it's like the corona on the sun. That when they saw it in the transmission electron microscope, they saw it that, oh, it's like the corona of the sun. So they gave it a name as like coronavirus. So the S protein on the viral envelope attaches to the cellular receptor ACE2 to enter the host cell. So this is like the lock and key. So the virus holds the key to the lock, which is the ACE. So when they attach to the ACE, they will be internally taken by endocytosis, and then the virus genetic material will be released inside. So this is the transmission electron microscopy of the coronavirus. So when viewed in the electron transcription in the electron microscope, the the virus that causes COVID-19 this is isolated from a patient in the US. You can see in here on the picture, it is emerging from a cell. And you can see the corona on the spike on the surface. That's why they gave the name coronaviruses. All right. Though inside the virion, which is a complete unit of the virus, is the blueprint of the virus, which is the positive sense RNA genome over 30 kilobase in length. A kilobase, 1 kilobase is 1,000 nucleotides. So this is a 30,000 nucleotide in length. This is the one of the largest in RNA virus. So this is a big genome. This codes for all the information it needs for entry, replication, and egress. So egress is the exit of the virus from the cell. So as you can see inside, there's a genetic material, and the outside of this virus is the membrane and the envelope and the spike. So the genome of the virus is unusually big, which is 30,000 kilobase, and they express into several structural and non-structural protein, or we call this polyprotein. So when the virus enters the cell, it releases its genome of 30,000 nucleotides, or 30 kilobase, and the protein of the virus will start to be synthesized. It's called, as Thomas emphasized, it's called translation from the RNA, which is the messenger RNA. So in the genome, there are the presence of a variable and conserved regions. So this is the whole genome of the coronavirus. As you can see here, this is 30,000. This in this case is 29,000, but it's around 30,000, including the 3' UTR. And this is the whole genome. So inside this genome, there are regions that we call variable and conserved region. This variable region, what I mean about variable is the nucleotide. Okay, sorry, where is that? Okay. So when I say variable region, the nucleotide can change, but the virus can tolerate those changes and can still replicate and continue its life. Okay, can still infect a new cell. So because the changes that is happening, which is what we call mutation on the nucleotide of the virus, is not that important. It can still survive with that mutation. It can still infect the host cell, and it can still make new genomes. But if the region that the mutation happened is in the conserved region of the virus will not survive that mutation. So scientists identify that conserved region in the coronavirus, and they are located in the RNA-dependent RNA polymerase in the E gene region and the nucleocapsid region. Okay, so but to better explain this mutation and the variability and the conserved, if you make a spelling mistake of the varnished, like in here, that's highlighted in red, you can still read the message. So it's still varnished. So that word will still survive. You can still interpret that you have to varnish something. So you can still do the job. But then if you change the spelling to V and T, which is varnished, to tarnished, the word will not survive. So varnish and tarnish are completely different words. For example, you are publishing a manual how to varnish an antique wood, and then the manual printed a tarnish on the title. That manual will not survive. You have to throw it away. It's the same as the virus. When it makes a mistake or a mutation on its genetic code on the very conserved region, the virus will not survive. Natural selection in the evolution of the virus will not allow it to survive, and then the virus will not survive, and it will not start to replicate. So that's how these viruses are continuing its survival and continuous mutation. So these conserved regions are ideal also for designing primers for RT-PCR. This is what the scientists do. They identify this, and they actually identify this in China and publish this a specific region, and they gave this to everybody else in the world. So they share this region to us, to US, to everybody else in Germany, And then they said that this is the conserved region, and then you can do the RT-PCR. Without this conserved region, we cannot do the RT-PCR because primers are this region, the primer development is dependent on the region that is on the conserved region of the virus. And also, scientists also identifies the antigenic region that stimulates the production of antibody by identifying the conserved region in the virus, and also for developing antiviral targets. So to simplify this previous slides that I am showing, that are the different proteins on the virus. Let's just draw a line. This is the whole genome, and there are 3 conserved regions that was identified. It's in the RDRP, in the envelope region, and the nucleocapsids. And specifically, this is the region that was identified. It's only like 100 nucleotide long out of the 30,000 nucleotide that is in the virus. So when they identify this, so this is the target that we use for the development of the primer for the use in the RT-PCR process. Okay, so now we go to the coronavirus life cycle. So when the virus encounters the cell, as I've said earlier, the spike protein on the surfaces of the coronavirus will attach to the ACE2, which is the receptor on the cell, and then it will start the endocytosis and will be enclosed into the endosome. In the late endosome, the genetic material will be released. After it is released, it will be translated, because the genetic material is a positive sense RNA genome, meaning it will be translated into protein directly. It doesn't need anything anymore. It will just hijack. So the virus is typical for hijacking the cellular components of the cell, the cellular machineries. So the first thing that they will hijack is the protein making machinery of the cell, which is the ribosome. So as you can see here, they hijack already the ribosome to start their translation. So the first thing that they will translate, that way the virus will make, is the RDRP. The RDRP stands for RNA-dependent RNA polymerase. So what does this do? The RDRP start making copies of the viral genome. So they make millions or billions of copies of these viral genomes, and then they make this genes copies, which is again translated into another kind of a protein, which is, as you can see here, will be translated into nucleocapsid, spike, membrane, and envelope, and of course the whole genome, which you need as the genetic information of the virus. Once you have all of this, it will then now attach to the membrane of the endoplasmic reticulum or into the Golgi apparatus, and then it will now carry this into formation of a new virus, part of the membrane or the envelope of this cellular organelles, and then they now start to egress or exit the cell. So this happened very, very fast, because the virus doesn't need anything else when it enters the cell. It just starts its translation. Okay. Now I will show you a slide. This is a crowded slide, but I will guide you through it. So this is a graphical representation of how coronavirus infects the cell. This is from Dr. David Goodsell. He developed this. So this illustration shows a cross-section through a cell infected with the coronavirus, such as SARS-CoV-2. So it shows a time point when the virus is actively replicating and the new virus are being created, as you can see here. Okay, so in this illustration, it actually shows that the virus is in the endoplasmic reticulum. This is the endoplasmic reticulum, and then start making the viral replicase. The viral replicase is the replicase and transcription complex. These are the RDRP RNA-dependent polymerase. So they will now start making copies, and then they will go to the double membrane vesicle of the endoplasmic reticulum. There are many RNA inside, as you can see in here, and start making new variants. As I earlier mentioned, they will start making viral proteins, nucleocapsids, the membrane, the envelope, and of course the copying of its own genome, which is the replication. They will now do this, and to create a new virion, and then the virion will bud out of the cell. This is now the mature virion. So why is now the COVID-19 so deadly? So it is deadly because it infects very fast, it replicates very fast, and when it infects, it makes billions of copies of its genome inside the cell, and this will now trigger antigenic reaction inside the cell and starts a cytokine storm. So when the cell is infected by a virus, it will communicate, and the interferon will now respond, and it's called the cytokine storm because there's so much virus in the cell and also in these pneumocytes. There'll be so much cytokine, and if the virus is destroying the cell, destruction, and there are cytokine, there will be inflammation. If there is an inflammation, there will be a fluid accumulation in the lungs. And if there's a fluid accumulation, the exchanges of the air, oxygen and carbon dioxide, will not happen on the capillaries. As you can see here, the capillaries. So the capillaries are with oxygen and the cells has carbon dioxide, but because of the fluid that is accumulating inside the alveolus, the exchanges of these gases doesn't happen. So because the virus is very aggressive on replicating, it's stimulating the production of this cytokine. So the cytokine is actually that is killing. Sometimes it's killing the patient because of the cytokine storm. Okay, so but then because although we have this virus is very deadly, we can actually defeat it. So we have to follow government guidelines, cover faces, social distancing, self-isolation, and keeping safe, and avoid touching any fomites. Fomites are anything that is contaminated by droplets. For example, your phones, the doorknobs, or the table that you handle, or anything, your gloves or your mask. This can be fomites. And avoid visiting crowded places. And of course, use appropriate disinfectants. When we say appropriate disinfectants, if the virus is an envelope virus, use a disinfectant that kills envelope viruses. If it is a non-envelope viruses, which is more stable, you need a more fast-acting disinfectant. For example, a phenol-based disinfectant can kill a non-envelope viruses. And always use our common sense, of course. You never leave home without your common sense. So, of course, you always need it every day in order not to infect any other people. So before I end my talk, I'd like to show some video on how can we combat this nasty disease. So the video will be shown shortly.
Washing your hands with soap is possibly the simplest way to flush pathogens down the drain. Soap molecules can interact with pathogens, such as coronavirus, leading to their destruction. Each soap molecule has two parts. The head is hydrophilic, which means it likes to interact with water, while the lipid tail is hydrophobic, which means it wants to get away from water to find similar water-avoiding particles. This is the basis of how soap works. When soap molecules encounter lipid particles on our skin, the tails aggregate around them to form spherical structures called micelles, creating a water-free environment. Similar things happen when they encounter enveloped viruses such as coronavirus. Envelope viruses carry their genome and supporting proteins inside a lipid bilayer membrane, with the proteins necessary for infection embedded in the membrane. In cells and viruses, These lipids are packed neatly into 2 sheets with the lipid tails facing inward. The lipid ends of the soap molecules are attracted to the lipids in the membrane. They take advantage of the presence of membrane proteins, which can perturb the neatly organized bilayer, to insert themselves into the viral membrane. If there is only a small amount of soap, the soap molecules only loosen the membrane. But with more soap, they begin to create micelles around membrane lipids. They are also attracted to hydrophobic amino acids in the membrane proteins, extracting them from the membrane. The structural integrity of a virus is essential for infection. So, washing your hands with soap protects you better from infection than washing with only water, and the longer you lather, the higher your chance of destroying the virus.
Right. Thank you very much, Dr. Ted Fajardo, for that very valuable lecture and detailed lecture about the basics of molecular virology. We now go to the last speaker for this session. The last speaker is a graduate of Bachelor of Science in Biology, Microbiology from Damascus University, Faculty of Sciences, Syria. She had his Master's of Science in Molecular Biology of Infectious Diseases from London School of Hygiene and Tropical Medicine, London, UK. She had his PhD in Molecular Virology at London School of Hygiene and Tropical Medicine, London, UK. She received several scholarships and awards. She published several papers in peer-reviewed journals. A research fellow specialist with over 10 years of experience in molecular biology, virology, and gene editing systems using CRISPR-Cas9, lentiviral, and AAV vectors for gene therapy and vaccine development, as well as over 5 years experience with medical microbiology. She an experienced scientist who achieved successful outcomes in leading and managing research projects. She's a postdoctoral scientist in RNA viruses, host cell interaction at Newfield Department of Medicine, University of Oxford, to talk about the fundamentals of PCR, real-time RT-PCR, and qRT-PCR. Let us all welcome Dr. Kinda Al-Sheikh Ahmed.
Thank you very much for the introduction, and hello all from London, UK. I'm really glad to be part of this significant event, and I wish I will deliver the information in a useful and simple way, because I'm going to talk about the basic and very important molecular biology techniques in every research and diagnostic lab. So I'm going now to start sharing my slides. Can you see me now, all of you? Can you see my slides?
Yes.
Dr. Kinder.
Okay, yeah. So I'm going mainly to talk about the techniques being used in nucleic acid amplifications. So the aim of my talk is to understand the principles of PCR, real-time PCR, and real-time reverse transcriptase PCR, in addition to qPCR and qRT-PCR. I'm also going to highlight the main components and steps of each technique. Later on, I will also focus on the advantages and application of each one, and I will determine which method which method are we going to use in order to analyze and visualize data. So first, what is the PCR? PCR is the polymerase chain reaction that was invented in the 1980s by American biochemist Carey Mullis, for which he won a Nobel Prize in 1993. This technique is widely used in molecular biology, and its main purpose is to rapidly amplify a very specific DNA region into million or billion copies. Like DNA amplification in an organism, the one that Thomas previously talked about, PCR requires a DNA polymerase enzyme, which we call it tag polymerase, that makes a new complement strands, cDNA.
Kinda.
Yeah?
Sorry,
Yeah?
can I interrupt? There's a sound.
Yes.
Can
There is a sound?
somebody work? Yeah, there's a sound like every time.
Is it okay now?
Kinda has to turn off the notification.
Hello?
Kinda has to
Hello,
turn
can you hear me now?
Yeah, there's a sound, yeah, notification sound every time. So it's interfering your talk.
I'm
Yeah.
sorry for that.
Can you turn off your notification sound?
Yes. I'm trying to. It's from Zoom meeting actually, so sorry guys, what should I do?
Dr. Kida,
Can
you
you
can mute your laptop.
Yeah, your volume on your laptop, just put X on it.
Is it okay now?
Yeah,
yes.
Okay, that's fine.
Now?
Yes.
Yes.
Okay, continue. Thank you, Kinda. Sorry.
Hello?
Yes.
Oh, there's still notification sound.
Yeah, because when I switch off, I will switch off everything. So this is the problem.
You can disable the computer sound.
Hala na siya. Narinig mo. Kay Aileen.
What happened to Kinda? While fixing the video of Dr. Kinda, can we ask some questions? Dr. Ted, can we ask the questions? I'll throw the first question. Are you listening? Can you hear me?
Yes, yes. All right.
You're right.
Can hear.
This is the first question. Either Thomas or Dr. Ted can answer.
Okay.
Hello, can you hear me now?
Oh, yeah.
Yeah, we can hear you.
Yeah. Okay.
And can you see my screen?
Yes,
Yes.
we can see your screen.
Okay, sorry for that, but there was technical problem. We tried to fix it. So I'm gonna, do you want me to start? I will start from the beginning just to talk about the PCR.
Yeah.
All right.
Yes.
So the polymerase is polymerase chain. Yes.
Your screen, share your PowerPoint.
Please share your PowerPoint, your slide deck.
Yeah, I am
Share your
sharing them.
Share your slides.
Yeah.
All right. Okay.
Can you see it now?
All right. Perfect.
Yes.
Yeah.
We're seeing
Yes.
your
Okay.
desktop. Yeah, that one. Okay.
All right.
Thank you.
Thank you.
Yeah. So polymerase chain reaction was first invented in 1980 by American biochemist Carey Mullis, for which he won a Nobel Prize in 1993. Then later, this technique was widely used method in molecular biology, and its main purpose is to rapidly amplify specific DNA gene into million or billion copies. Like DNA replication in an organism, and this is Thomas previously talked about, PCR requires a DNA enzyme, which we call it tag polymerase, that makes a new complement strands of DNA using existing strand as a template. Since the invention of PCR, it's been elaborated in many medical and clinical researches and is now commonly used for a wide variety of applications, including biomedical and criminal forensics. So what is the principle of PCR? In PCR mixture, there are 5 essential reagents should be added. First, the DNA template, in which the region we are interested in amplifying is present. The second one is the primers, the forward and reverse primers, which are short sequence of nucleotides up to 20 nucleotides in length. And this is to provide a starting point for DNA synthesis. These primers should flank the target DNA region. The third one is the DNA polymerase, which is an enzyme that amplifies DNA during the cell cycle. In PCR, this DNA polymerase can tolerate high temperature up to 96 degrees centigrade. And this temperature is necessary to separate the 2 complement strands of DNA in PCR tubes. Other reagent that is important in the PCR mixture is the 4 types of nucleotides, been also talked about before, in which DNA polymerase will grab the complementary nucleotide and attach them to the 5' and pairing them with the other template DNA. The last reagent that it's important to be added in the mixture are the buffer and also the magnesium chloride. These important in order to maintain the right pH and provide necessary ions for the enzyme in order to work. And of course, all this reaction will be carried out in the automated thermal cycler machine. Then what is the principle of PCR procedure? A typical PCR procedure that takes place in PCR machine that I showed in the previous slide consists of a series of 20 to 55 repeated cycles with consistent temperature changes. In each cycle, there are three steps. The first step is the denaturation. In this step, two strands of DNA are separated or denatured to provide the single strand template for the next step. And usually the temperature ranging between 90 to 96 degrees Celsius. The step after that is the annealing. Annealing temperature range between 55 to 65, and this is depending on the primers. The reaction should be cooled so the primer can bind to their complementary sequence on the single-stranded template DNA that generated by the denaturation process. And then the purpose of this primers is to prime polymerization of the DNA. The last step is the extension, in which the reaction temperature raises all up to 72. So the tag polymerase extend the primers, synthesizing new strands of DNA. The addition of each nucleotide always start at the 3' end. These three steps will be repeated several times, and the number of cycle copies will be doubled by the end of each cycle, thereby increasing exponentially. And generally, PCR reaction takes between 20 minutes to 4 hours, and this is also depending on the length of the DNA region that we are interested in amplification. Then, okay, now we have we decided which part of the DNA are we going to amplify, and we did perfectly PCR mixture. So how are we going to visualize the products of PCR? The results of PCR reaction are usually visualized using gel electrophoresis. In this technique, fragments of the DNA are pulled through gel matrix by an electric current, and it separates DNA fragments according to the size. So the DNA migrates into the positive electrode, and this is because the DNA is negatively charged due to the phosphate group. Then DNA bands or fragments again can be visualized in stained gel with ethidium bromide or cyber green. I'm going now to talk about another type of PCR, which we call it reverse transcriptase PCR. So the reverse transcription is the generation of complementary DNA, what we call it cDNA, from the RNA template. And the reverse transcriptase PCR is a variation of standard PCR that involves the production of cDNA from RNA template using the reverse transcriptase enzyme. Then this cDNA used for exponential amplification by normal PCR or by real-time PCR. The RT-PCR is a very common technique in virology diagnostic. And when it combines with real-time PCR, the level of messenger RNAs is quantified in a specific sample. So what is the concept of this reverse transcriptase PCR? First, to perform this technique, RNA sample is extracted from a pathogen. And here I'm going to give example of the SARS-CoV-2, or it might be any other bacteria or even from cell or tissue. So in this technique, and in addition to all standard reagents used in basic PCR, we have to use the RNA as a template, and also the RT transcriptase polymerase. The reaction mixture is then heated to 55 degrees Celsius, which enabled the production of the cDNA from the RNA template. And this cDNA then anneal to one of the primers, leading to the first strand synthesis. Primers used in this example, as I mentioned, as specific to the SARS-CoV-2 conserved sequence, the domains that Ted discussed before, and this will then prime copying the SARS-CoV-2 RNA into cDNAs. Later on, in this slide, I'm talking about other types of PCRs, which are qPCR or quantitative PCR, and this is this technique used in order to detect, characterize, and quantify nucleic acid using the DNA as a template, while the qRT-PCR use the RNA transcripts, in which RNA transcripts are quantified by reverse transcribing them into the complementary cDNA. The RT-PCR, this technique that my other speakers later on in later sessions will focus and talk more in details, is a method for just detecting the presence of the specific genetic material, RNA, from a pathogen in a real time. So it's just for the detection of the presence of this genetic material, not for the quantification. So during the pandemic time, this technique is most widely used for the detection of coronaviruses, and there are many countries still need support in setting up this technique. So what are the advantages of the real-time PCR? This technique is the most powerful and sensitive analysis method because it allows the generation of accurate quantitative data. And this is because it has a fluorescent reporter molecule, for example, FAM or ROX, that monitor successfully the accumulation of PCR product. Thus, increase the dynamic range of nucleic acid detection. And this is because it's sensitive technique. The detection of RNA molecules is down to 1 copy. And since a fluorescent reporter is used, the accuracy to detect smaller fold change is also increased. And as a result, the whole throughput is also increased. So how this real-time PCR works? Here I'm going to talk about Tagman assay, which is one of the earliest introduced technique in real-time PCR for both quantification and also detection of nucleic acid. So in this method and during annealing process, a primer and a probe anneal to its complementary sequence. The probe is usually located between the forward and the reverse primers. This probe consists of a fluorophore reporter, such as a FAM and ROX that I previously mentioned, and also it has a quencher, like blackberry quencher, that inhibits fluorescence due to their proximity to each other. In the presence of in the process of polymerization, DNA polymerase starts to extend primers, moving towards the probe. Then endonuclease activity of the tag polymerase cleave the first nucleotide of the probe, separating the FAM from the quencher and resulting in degradation of the probe, and fluorescent starts to emit. This reaction will happen again on the second cycle, and the measurement of the fluorescence will be increased and detected by optimal detector in a real time. So I'm going to talk a little bit about the measurements of that takes place in our RT-PCR, and this is briefly because other speakers will focus on. So in real time PCR, and similar to basic PCR, there are three phases of amplifications, which are exponential, linear, and plateau. And in in our RT-PCR, we focus on the exponential phase, which provides the most precise and accurate data for detection of a specific nucleic acid. During RT-PCR, the instrument calculates the exponential copying of the amplicon per cycle. And there are two important values should be taken into consideration in the real-time PCR for calculation and for also data analysis. These two values are the threshold and also the CT value. The threshold give us the level of detection at which a reaction reach a fluorescent intensity above the background, while the CT value is the number of cycles at which the sample reached the set threshold. This value is used in an absolute or in a relative quantification. So I just want to mention briefly that a quantitative PCR is the same technique as our RT-PCR, except that in qPCR, you will have the exact number of amplicon compared against the standard. Later, there are other modified technique, let's say, from the PCR, which is the multiplex PCR. In the multiplex PCR, we use it in order to amplify multiple targets, and this is in a one single PCR experiment. To do this assay, we use multiple pairs of primers, forward and reverse, in each reaction mixture. The main goal or purpose of this technique is to save time and effort in comparison to the normal PCR. There are two types of the multiplex PCRs. Single template, in which the genomic DNA is used in addition to other set of primer pairs, or multiple templates in which multiple genomic DNA and several set of primer pairs are also used. So in summary, we covered the principle of each biological technique that been used in different research and diagnostic labs. I also highlighted the components and the procedures of these techniques. I focused on the importance of the applications of the real-time reverse transcriptase PCR, and this is because due to its role in the diagnosis of pathogen infection and viruses in particular. And thank you very much for your listening.
Thank you very much, Dr. Kinda, for the very detailed explanation of what is PCR and the different formats of PCR. We now go to questions and answers, answering the questions. Now we would like the participants to place your questions. I'll shoot the first question, and all the three foreign speakers may answer. The Philippines is lagging behind in terms of adoption of molecular techniques and molecular-based techniques for testing. What can you recommend so we can catch up with the developed countries? Maybe Dr. Ted can start.
Okay. Hi. Thanks for the question. So we are lagging behind because we don't have that much budget for or support from the government. So it is before there's also no foresight from the government. So we don't have a virology institute in the Philippines to focus on virological studies or molecular biology. If we had this before, this would not happen, like we are lagging behind, isn't it? So what we have is a full support from the government, and also constant professional development from the teachers. The teachers can teach proper molecular biology and virology or molecular bacteriology. They should also be updated, the teachers, the medtech teachers, and of course funding from the CHED or from the DepEd to provide training for these teachers. And when it comes to workplace, we should have funds from the Department of Health, if you're from the government, to provide this the materials that you need, and of course training. And if you're from the private sector, then I cannot answer if the private sector can provide for that. So yeah.
Yeah, thank you, Dr. Ted. May I ask Dr. Kinda if she has some suggestions? Dr. Kinda?
I think, yes, I think it's very important to have collaboration, international collaborations with other universities. For example, several universities in UK or even in the USA, in order to support the establishment of specific labs for research, and also to do training, proper training, and proper lecturing.
Yeah, thank you very much for that. What about Dr. Thomas? Any suggestions, any comments?
I can just add to all this that even though we missed the sort of time window to prepare, and this kind of globally, we missed the time to prepare for this pandemic. Now that we are more aware of it, it's still now time to really focus and implement all those necessary changes. So if it happens again, and everything suggests that things similar to this may happen again, we are better prepared next time. So let's learn from this pandemic.
Yes, thank you very much. Now we go to the next question. This is specifically for Dr. Gigi. What is the biggest challenge that we need to overcome to reach the target of the government for COVID-19 testing?
Okay, currently we are ill-equipped in skills, knowledge, and basically the lab facilities available to hit the target. So we need more proficient RMTs to perform this COVID testing, so that we would be able to help the government in reaching the target.
Thank you. Dr. Ted, do you have any comment about that?
Oh, sorry. Am I okay? So to fully reach our the capacity, the target of the government, of course, we need like trained personnel. Like this is what we are doing now, but this is I hope it's not too late. So we are providing a capability to people who can do the job, and also, of course, we need more budget for this. So they are targeting tests. Are they giving the proper budget for that, like to buy primers, to buy probe, to buy the machines, to buy, for example, the proper methodology to, for example, the extraction method. What is the extraction method? Are you using a conventional or a machine? Like, in order to do more, you have to automate everything, isn't it? So
yes,
we need a lot of a fund for that.
Yes. Thank you. Thank you.
In
Now,
addition to that,
yes.
Dr.
Yes.
Okay. In addition to that, I would like to give importance to what Thomas suggested. We have to learn from our experience. So from this time on, we have to be aggressive in learning new technologies And so just like here that we are doing, we are having our free webinar series. I hope we could come up, the PAMET could come up with more information sources for and trainings for our members in PAMET. So I think we would be able to come up with better solution and address the target of the government.
Related to this, the medtech should be competent and proficient in the molecular biology techniques. It cannot be learned overnight. The knowledge and skills have to be developed. How do you propose to enable the medtechs to be proficient? I know you mentioned about this webinar. Maybe there are more. Can you suggest? Anyone can answer.
It's difficult now because to start the training and for the medtechs to become proficient, we need equipment, we need training facilities, we need the right methodology. So once these are all in place, I think we should all do this first, provide everything, and then the trainers can come. And then once everything is in place, then we can do it like this. We are starting it now. So I hope somebody in the government can continue it and then provide all the facilities that we need for the medtech to be better equipped to test for testing.
All right.
Can I add something also?
Yes, Dr. Arkinda.
Yeah. I want to say that, of course, learning process is long, but there is always a start in every step. So, and this what you are doing now, it's a very, very important step. Other way of learning, and I found it recently they do it in London to continue teaching undergrad students in science. They call it virtual scientific labs. So they explain all the experiments virtually, and it's been and someone just do the experiment in front of all students. So students at least get an idea how to perform the experiments. So when they go in person, they have a strong background about how to do this technique itself, and they just want to practice it. So this is other way maybe if you can launch it later or think about it during the next time.
Yeah, thank you. Anyone else?
Dr. Lila, can I add something to that as well?
Yes, Dr. Mirna. Yeah.
So I think it would be sorry, but that's okay. I think it would be good if we can add that to the curriculum of the medical technologists in the
It's already included for the new ones.
Like it's quite similar, as I'm part of the Department of Veterinary Medicine, and during, I think way back 10 years ago, we did have PCR lectures, but then we were not able to do it in practical. So I think it would be necessary that in addition to theoretical aspects, they should be able to do it hands-on, they should be able to practice, and universities should have it, and they should be doing it hands-on. Yeah.
Right. It's already in the new curriculum of medical technology, but then those already working are not exposed to such technology. So that's why we're having a hard time.
Yeah.
There is one question here. Would this e-learning session suffice to comply with the DOH requirement for training the RMTs for those intending to put up molecular lab? I don't think so. Somebody else can answer, but I don't think so. Yeah.
Dr. Laila,
yes.
I think this one is a stepping stone, okay,
Yeah.
for those who do not have any background yet to in molecular biology and virology, and also this would help us also in the next 2 sessions that we will be having, like section 2, the real testing procedure in COVID testing, and then the session 3 would be the demonstration. All of these are not really enough, but this would help them get an idea on how the flow or the proper techniques and biosafety measures that they need to consider.
Yes, and also to remind everyone that there should be a proficiency testing for the analyst. So you will be given a known and you have to pass it before you can be qualified or accredited. The lab will be accredited.
Yeah, if I can just add something to
Yes,
this.
Dr. Laila.
Yes. Yes,
Sorry.
Thomas. Yes, Thomas.
I just wanted to emphasize that all molecular biology techniques, especially in terms of diagnostics, are very challenging, but also very demanding, and there is no room for any compromise. So if you want to use it, we need to make sure that we have state-of-art technology, we have like experienced staff that can fully understand what they do, and they've been doing it for long enough after a precise training, because if you can often hear, no test is better than a bad test. Like the worst we can deliver is a diagnostic tool of low accuracy, because that will be actually more harmful than helpful.
Yeah. Thank you, Dr. Thomas.
Dr.
Yes.
Okay, if I may add about the proficiency to this COVID testing must be provided by the NRL for infectious disease, which is the Research Institute for Tropical Medicine, so RITM of the DOH.
All right, thank you. There is another question here. The World Bank gave loan to the Philippines for the PCR machines, but they procure PCR machines from China. Any comment about the PCR machines made from China?
Okay. Oh, who's
I would like
going
to answer.
Okay, Myra.
I think all of our, it could be possible that any machine from China are not as good as the machines that are made in Western countries. But also, I think we should understand that any machine, as long as it's a PCR machine, should work as long as we know the basics of every machine. It is not necessary that we should have Thermo Fisher made machine or a Roche machine or reagents from QIAGEN. It is definitely possible to use reagents from China, machines from China, as long as you know what you're doing and as long as you know how the basic principles are working, how the reagent should be working. And I think you should be able to do your job and you should be able to diagnose as well, just as good as when you're using other machines.
Yeah. I'd like to add to that. So yeah, it's not actually the machine, it's the technique. So RT-PCR is the detection of a fluorophore being emitted while the PCR is happening, as what Kinda is saying. So this emission of a fluorophore is happening while the probe is being hydrolyzed. So any machine can detect that. If they design a machine that can detect it, you can use it as long as it can detect the right number of a fluorophore in real time. Then they can do it. Also, the PCR machine, you can use any PCR machine. So it's not about China. It's about the machine that if it can actually detect the fluorophore, can give you the number, can give you the right CT value, then it's good.
Yeah.
And also
can I add something and emphasis on what Dr. Ted was talking about? Yeah, I also agree with him that it's not the machine itself, but you have to pay attention more to the technique and also to the reagents you are using. So you have to have good reagents in order to get the correct fluorescence. And also the technique is very important.
So we will discuss this more in detail on the next topic, which is the RT-PCR itself, the methodology, interpretation, and the preparation of samples.
Yes, many of the questions should be in the
On the
second.
on the
Yeah,
second
I
one.
have one last question. Okay.
Can I interrupt you, Ma'am Lai? I have a question
Yes.
here.
Yeah.
So it says here, I was reading it. It says, can the overproduction of cytokines be the reason for sepsis development in some COVID-19 patients?
Okay.
So that was the question. So well, yeah. So sepsis development is it's depends on the sepsis is bacteria, isn't it? So when sepsis comes, it's a secondary infection. So it's secondary infection from a bacteria. It's not from the virus that maybe the patient is dying from a secondary infection because the immunity of the patient is down because of the cytokine storm. Could be that is the reason, or could be that because of the cytokine storm, the immunity of the patient is very busy fighting the virus, and then the next thing to come is the bacteria, and this secondary infection will kill the patient. It's possible. Yeah.
And can I add to that, please?
Yeah.
So actually with coronavirus infection, especially with COVID-2, it is possible that there would be a cytokine storm during the infection, but there is no very strong evidence yet, but it is possible.
Yeah. Everything in the SARS-CoV-2 research is rapidly developing. So what might have been proven now could be wrong tomorrow. So we don't know that yet. It's rapidly developing science.
Yeah.
Okay, you can continue, Ma'am Lai. Sorry for interruption.
All right, one last question. I don't know if this will fit the in the second, but I'd like to pose this question about the difference between the PCR, the RT-PCR, and the gene expert. As reliable, is it as reliable as the PCR, RT-PCR we're using?
Gene Expert is a brand, isn't it? So as long as it's the principle that the Gene Expert is using is the same as RT-PCR.
It uses a cartridge. It's automated.
Yeah.
So they get the sample and then
which company is making Gene Expert?
Cepheid.
Cepheid, yeah, Cepheid. So it's a brand, like what we are talking about, Kinda and me. It's not about the brand or the machine. It's about the ability to detect what it has to do to detect.
Yeah, correct. I agree. It's just that this is automated compared.
Yes.
Yeah. And
Dr. Laila, can I add something?
Yes.
So sometimes there are master mixes that are just compatible with certain qPCR machines. So it is very important that we are aware of that. So this would be in the next session anyway, but it is important that you know that your master mix is compatible with the machine that you're using, because it will definitely affect your result. Just for example, if you use a passive reference that is not compatible with your qPCR, it will totally affect your result and you will get no amplification.
Exactly.
Yeah. So most of the questions belong to the second session. So advise the audience, the participants, to keep tuned to the second session. It's all about the technology, the techniques.
Yeah. Can I add to that? Yeah, because most of the questions that I read is about RDRP, E and EG, and specificity of the target. These are covered in the second session.
Yes, thank you very much. Thank you very much to our 4 speakers for this session. Yeah, I'd like to give the synthesis for this session. The first speaker gave the topic about the COVID-19 setting in the Philippines. She focused on the current situation for COVID-19 testing, including the testing platforms in our country, the regulations involved, in the performance of the test and the challenges involved in the performance of the test. She specifically mentioned about preparing the battle. The medtech should know our allies, should prepare the weapons, and should know how to set the lab. We should understand the enemy, and our enemy is the SARS-CoV-2. So, but we should also understand our limitations. So she mentioned that there are 30 lab accredited with 3 medtechs per lab. I know the DOH aim is to increase the testing facility. So there will be more. But then we have backlogs. That's the problem. Many, so we have to win this battle. Risk assessment in each lab is needed to ensure biosafety and biosecurity. The second speaker talked about the fundamentals of molecular biology. He mentioned about the processes of replication, transcription, and cell function. He mentioned the central dogma of molecular biology, where genetic material is transcribed into RNA and then translated into protein, and current revisions in light of emerging novel roles for RNA was discussed in details. So the role of the field in the laboratory testing was given emphasis. The third speaker, Dr. Ted, discussed the viruses at the molecular level, which involves the analysis of chains and chain products of viruses, and study their interaction with the host, the human, particularly cellular proteins. Emphasis was on coronavirus, the structure and the role of the said structure in molecular-based assay. He said, we are all part of the virus. A portion of the virus genome is actually in our genome. So he explained very clearly the structure of the virus genome, SARS-CoV-2 in particular, the life cycle of the coronavirus. Yes. He said it is very deadly, but we can defeat it. Just follow guidelines, avoid touching the fomites, social distancing, avoid crowded places, use proper disinfection, and use common sense. And the last speaker talked about the PCR, the different formats of the PCR, the real-time RT-PCR, and the quantitative RT-PCR. She discussed the concepts of amplification of DNA using the PCR technology. Different formats of PCR, from conventional to real-time to emerging formats, were included. The role of PCR in the laboratory was given due emphasis. Factors that affect the PCR run, the troubleshooting, and analysis were also discussed. So everything about PCR technology, the process will be discussed in the second session. Thank you very much for this.
Thank you.
Okay, thank you.
Now, yeah, thank you very much. We will go to question and answer. We're finished with the question and answer. Now, how to claim the e-certificate. The succeeding session will use the same link in the first session. Answer the webinar post-evaluation and post-test at any of the links to be flashed in the succeeding slide. If a link is not working, you may use the other links instead. You need to garner at least 60% score in the post-test to merit an e-certificate. The e-certificate will be sent to your registered email address. The link will only be open for the next 30 minutes. So you have to answer it within the next 30 minutes. Okay, are we ready?